So what I'm going to present today is the paper on the modified create intervention that I did in my genetics course. It's an upper division genetics course, and how it improves student outcomes both in the cognitive learning, so related to uh, content learning in genetics, as well as affective outcomes, which includes motivation, interest, and community in the classroom. And so in fact, want to talk a little bit about how I got into biology education research to start with. Most of my formal training is in biochemistry. Both, actually, all three of my degree, right, at all three stages were in biochemistry. But before, after undergraduate and before my PhD program, I actually went and taught high school for a couple of years, which was really at least for me, a really formative experience, as being, a first job out of college and being responsible for a large number of students and also keeping up with actually teaching the content, right? And then after my PhD, I did a short postdoc in biochemistry and then did a much longer postdoc in learning sciences, which is an interdisciplinary field, trying to understand how people learn and how anyone learns anything at all. And I was really fortunate and privileged to have that second sort of postdoctoral phase where I was then able to explore a lot of research interest in biology education research, but also have the opportunity to attend and participate in a number of conferences, many different conferences, from the ASM conference on undergraduate education to SABER, the Society for Advancement of Bioeducation Research, but also broader meetings such as the National Association for Research and Science Teaching, the American Education Research Association, which is all education from birth till death in any way, shape, or form, as well as the International Society for Learning Sciences, which is the society for this particular interdisciplinary field. And then I've been in my position since 2014 at UC San Diego. Currently, I'm an I recently became an associate teaching professor in cell and developmental biology, and I'm also affiliated with our PhD program in math and science education. I also want to put in a plug for ASM, has a discipline-based education research course that's been running since 2017. I think we're in a bit of a hiatus because of the current global situation. But I think, if you have more questions, I'm happy to answer them. But I think Rachel would probably have more information on the course and the future of the course as well. So to start, right, I actually want to talk a little bit about this particular teaching strategy called CREATE. It was originally developed by Sally Hoskins at the City College in New York, where students work in small groups and to really dive in, this is a deep dive into the primary literature. So in the original CREATE strategy, in a course, students would read 3 to 4 primary literature papers for the entire semester. So this is, quite a bit smaller number than you might imagine for a primary literature reading course, right? But the idea here is to engage students not just in a cursory look, right, of each paper, but really dive in depth into thinking about what are the questions, what is the biology going on, what are the methods and techniques and technologies, how do you actually do the experiments and look at the data and draw a conclusion from the data, and also think about what the next experiment might be. And in fact, in the very original CREATE strategy, the papers are connected. They are usually 3 to 4 papers by the same research group. So students are able to, design the next experiment and see, right, whether their proposed experiments and their proposed questions were in fact the questions that the researchers pursued after that. So, it was originally meant to be a combination of both an upper division course that's focused on primary literature reading, but also meant to be able to use as a way to teach fundamental concepts. It was actually, in fact, I think that the first version of CREATE was done in an upper division cell biology course. It wasn't meant to be just reading primary literature. It's the standard cell biology course that was taught at City College in New York. And what was really interesting about this method is that not only are we engaging students in that critical thinking and analytical reasoning, right, with the primary literature, but they're seeing the course content in a contextualized way that is, situated in how discoveries were made, right? And so another, that's one advantage. And another advantage is that, in a standard course, we often, if you look at the textbook chapters, for example, right, we go from one chapter to the next. They may or may not be connected. In fact, I teach genetics, right? And genes and all of these things happen, for a good 10 to 12 chapters in the textbook before we learn about DNA, right? And then by the time it happens, students don't even quite sometimes, remember that chromosomes have DNA because they're so, they're 10 chapters apart in the book, right? So the idea here is to really engage students in the learning process where they see, right, different concepts come together in one place. And because it's iterative, right, we're doing a second paper and a third paper, then they in fact see the concepts multiple times in the course, right? So this is pretty in-depth and involved strategy, right? And I just want to sort of lay out what it means, right? So CREATE actually stands for consider, read, elucidate, hypothesis, analyze, interpret data, think of the next experiment. It's quite a mouthful for an acronym, right? But essentially it's taking students through the paper one step at a time, right? So consider, what the questions and the concepts and the topics are. Read the article to really extract bits and pieces of the article, not the whole article, right? But bits and pieces of the article to really define, what are the words, what are the concepts, what are the methods. It involves a lot of, rewriting, rewording in students' own words, generating concept maps to represent connections of information, and creating diagrams for how different techniques and experimental methods work, right? And then, figuring out the hypothesis, because oftentimes that's implicitly stated, right, in the paper or in the figures, and then coming up, with a conclusion based on the data, and as I said, right, thinking about the next experiment. And so the challenge for me when I started thinking about, teaching my genetics course with this CREATE strategy is that I'm at a large public university where, we have, I don't know, somewhere between 10 to 15 sections of genetics taught by many different people. Each has, a few hundred students, right? And so then the question is how we can actually do this in a more standard curriculum, right? Because students do continue on into other courses. In fact, on my campus, genetics is prerequisite for a lot of other courses, and it's often actually the first upper division course that students would take after introductory biology. So part of the challenge that I wanted to take on is how do I then create, well, that's not a good word. How do I develop a course, right? A genetic course and more of a standard curriculum that brings in those CREATE elements and yet still allow us to have the sort of connection and semblance with all the other sections that may be, taught by many different people. And so instead of, right, going through 3 to 4 papers completely, I decided to pick and choose bits and pieces from many different papers. And instead of doing 3 to 4 papers or stories that are connected, I created 4 different modules. Some of them are more related to one another, right? But essentially there's 4 separate stories, right? So first is about DNA forensics, right? So, how can we use genetics to, find elephant poachers or reconstruct family trees, and particularly in this case, the family tree for King Tut, right? Then we went into human diseases, biodiversity, and human genetics, right? So each of them is a complete module that has its own sort of connections within itself. And then I drew in bits and pieces of, a few different primary literature papers to support the learning of each of those modules. And in the middle column, right, what we see is the genetics topics, right? And again, to really recreate some of those moments in the original create method where students are bringing together multiple concepts in one place, but also seeing the same concepts throughout, right? So in this case, in the first module, it's bringing together, this is the very first part of the course, right? So it's revisiting classical genetics like, alleles and Mendelian inheritance and pedigrees and things like that, but doing it from the molecular aspects. So bringing in all the molecular genetics, right? Using molecular markers, thinking about polymorphisms and meiosis, but also broadening it to population genetics and probability and statistics, right? And many of these concepts then come back again, because when we talk about human diseases right now, alleles and meiosis and all of those things come back, but now we add on additional topics or branch out to additional topics as well as we go through those 4 iterations. And so I want to point out, right, so the course, because, right, we are doing this sort of, combination of what might look like the original create method, but drawing in multiple papers, right? So what we did is actually, we did a number of different activities. And what I'm showing here is actually one class session. So the x-axis is the time. So these were, 80-minute classes, right? And on the y-axis is actually the decibel volume of the classroom recording. So this is a particular method called DART, Decibel Analysis of Research and Teaching, developed by Kimberly Tanner's group at San Francisco State. Essentially what it's doing is using the classroom recording to determine if a single person is speaking at the moment, or if multiple or many people are speaking at the moment, right? So it turns out when I'm speaking, right, one person speaking, so like I'm speaking now, I actually breathe in between, words and things like that. So the decibel volume, right, goes up and down pretty, widely. But when many people are speaking, right, those gaps actually, right, they fill in one another's breathing gaps, right? And so then what you see is a consistently much narrower and higher decibel volume. So that's how the algorithm, it's a machine learning algorithm that sort of figures out, whether it's one person speaking or multiple people speaking. So in this particular class, right, this is one class session, where we spent a lot of time focusing on data analysis. So there's an introduction of what we're doing. I did quite a bit of talking to introduce what's going on for the day. Student dived right in to analyze a piece of data. And then we had some report out and sharing. And so that's actually some of those. Single voice was actually individual students talking as well. And then we took some time and went back to actually look at more data. And then there's a summary lecture after that, right? And in other classes, right, because we are trying to also really, cover the main content of what would look like a genetics course, right? So I think this is the day that we talked about the basics of recombination before we went into a paper that did genetic mapping. So here is actually mostly a lecture interspersed with clicker questions, which is marked as CQ here. And what you also see is the CQ tended to have, they came in pairs. So there were actually 4 clicker questions. Students, talked and voted once, and then I gave a little bit of feedback, and then they talked more and voted again. So sort of in the classic peer instruction method, that's kind of the Eric Mazur kind of peer instruction method from physics, for example, right? So just to point out that there were sort of different kinds of activities going on, which is why we called this a modified create intervention. And so this is sort of the main data that I just wanted to show. So there were sort of 3 other genetic sections that were happening in the same quarter. And they sort of came in, different course sizes because we had different structure of courses. And so what we did was we gave students from all these different sections of the course a concept test or a concept inventory. So CI score is the score of the concept inventory at the beginning and at the end of the course. And so the bar graphs here are showing student scores on average with the standard deviation, right? How they did at the beginning and at the end of the course. What's at the bottom is the effect size. So ES stands for effect size, and it's the number of standard deviations that students did better at the end of the course compared to the beginning of the course. So basically the difference for post minus pre, and then normalized or divided by the standard deviation. So what we see here is that students learned, students performed about 1.5 standard deviation better at the end of the course compared to the beginning of the course. For the create version of that genetics course, whereas the other 3 sections had had smaller effect sizes ranging from 0.46 to 0.87. And what's super interesting is, and I particularly want to point out, is that we also asked students their perceived learning, right? So all the other courses were in lecture format, but they also had clicker questions as well for the most part. And when we asked students perceived learning on a Likert scale, so strongly disagree to strongly agree on a 5-point scale, students actually predicted about 0.5 point or more less learning in the create section of the course compared to the more classic lecture and clicker lecture courses. And so just want to point out that sometimes, right, students may be perceiving this as really challenging, right? Because we are challenging to learn and challenging to learn deeply. And they may feel that because it's challenging, they may feel that they're not learning as much. But in this case, in fact, they are, right? And so I just want to sort of close here and finish the formal portion of the talking. And I know I can't see the actual chat screen, but I see a little blinking that says now there are 10 chat messages. So I'm going to actually not stop sharing the screen, and then maybe we can have have a more of a conversation as well.
Great, thanks so much. Okay, well, for the first question that we have for you is, can you talk a little bit more about the machine learning that you did for this? That's quite novel to me. And what gave you the output, and how can someone go about using that in a study of their own? You're on mute, Stanley.
Sorry, I'm very good at muting myself and then forgetting about it. Sorry. So anyway, so the machine learning algorithm is called DART. I'm going to paste a link in the chat so everybody has it. It's actually, I did not have to do much. It was developed by Kimberly Tanner's group at San Francisco State. They have a website. Essentially, you drop in the audio file. It has some requirements of what the file needs to be. You drop it in, it goes through their algorithm, and then they give you the output of essentially what I showed you. And then I just shaded the region and added, my own annotation. But it tells you which region is single voice and which region is multi-voice. So I didn't have to do much other than putting the audio file onto the website and then sort of getting the output. So this is all Kimberly and her team's work.
Is that a free software?
It is. So so if you go to the website, you can set up a free account and you can drop in the file and it will give you the output.
Thank you.
I mean, so there are some complications. So like if you're in a big lecture hall and you have one of those lapel mics, it might not work because the lapel mic is actually designed to focus on the voice of the person and then minimize ambient noise. So it actually doesn't record the multi-voice talking as well. So in my case, I actually happen to have my campus does podcasts, classroom podcast, and in the classroom that I taught in, they had a ceiling mic to record the podcast. So that was actually really good for the DART algorithm.
That's good. I've heard of a lot of classes in the adaptation to hybrid learning. They have ceiling mics these days.
Yep.
That's good to know. A question from a participant: do you have the student read the paper before coming to class, or do they read it during the class? Talk a little bit about what you had the students do for homework prior to class.
Yeah, so it's a little bit of both. I don't actually give them the whole paper because then students tend to look for answers in the paper instead of processing the bits, the information on their own. So for example, right, if we're looking at a figure, they really just get the figure, right? Because in my experience, and I imagine others have the same experience too, right? That if you give students the whole paper, they would then actually look for the author's conclusion, right? Instead of thinking through the data and coming up with the conclusion on their own. So sometimes the homework may be, just read the introduction, right? And I would say, here are 5 keywords from the introduction, and you can add, you should add at least 3 more or at least 5 more, but you can add as many as you want, and then generate a concept map from the introduction. And then when we come to class, students would then compare their concept maps in small groups and then regenerate I guess a quote unquote larger one that synthesizes, a few concept maps together. And then we would sort of show them, show a few and talk through them together. So that's one way. Or, I might ask them to, look at the figure and annotate the figure in a way that, writes, marks, sort of annotate the figure and mark down, what is the x-axis and y-axis. So what are the researchers, right, changing, and what are the researchers actually measuring or observing, maybe circling and highlighting this part of the figure, like this the comparison I need to do to answer the research question. And again, so some of the homework is individual work like this with just very small bits and pieces of the paper. And then when we come to class, we actually compare and talk through what they've worked on.
Thank you. If appropriate papers were selected, do you think that this approach could be used in a 200 level course or even an introductory level course?
Yeah, so I would say the genetics course that I taught is probably what would be counted as a 200 level course, because it's, as I mentioned, it's typically, in fact, the first upper level course that our students take after intro bio, and then it's prerequisites for a bunch of other upper division courses. And then so in fact, the create strategy, over the years, Sally and other colleagues have adopted it to introductory courses. They've done it in freshman seminars. There's quite a bit of existing papers that people have selected for different courses and at different levels. The table that I showed for what CREATE stands for, that actually came from an American biology teacher perspective piece that Sally wrote, and that was in fact geared towards high school teachers to consider using this strategy for news article instead of primary literature, where students are engaging and dissecting information in news article and thinking, well, if this is the claim, right, that the news article is saying the scientists make, like, what kind of data do we need to support that and things like that.
Staying on the topic of what you've done, is there a textbook with your genetics course that you use this intervention with? And do you have regular assigned readings?
In this case, let me try to think. We did have a textbook because we all had the same textbook across the 10, 12, or 15 sections of genetics. So I also did a lot of, just like I was pulling out just little bits and pieces of primary literature paper, I did the same with the textbook. So, before we talked about recombination, students, I think, read just like, that very small portion about recombination. And then we worked through a bunch of problems in class. And then it's still tied into the main story of the module, which is then a paper where the researchers did recombination mapping to or QTL mapping to figure out where the genes are that's responsible, or the loci are responsible for the traits. So it's a little combination of both.
I was just wondering, how often do you use the primary literatures in your classes? Is it weekly, a couple times a semester?
So in the course, it was essentially probably weekly and probably most of the days. So even for the clicker lectures, that's trying to get at just fundamental genetics concepts from the textbook. Some of the clicker questions are still primary literature based. So I would bring in figures and analyze them together through clicker questions instead of the more involved sort of create discussions.
Yeah, I'm thinking if I were to use this in an upper level course, I would have one of my students in prerequisite courses to have had some experience reading primary literature. Did your students have that, or did you have to spend a lot of time helping them develop the skill to read the literature?
I think part of it is developing the skill, but I imagine, right? Because if I think back to my own graduate experience, right, in graduate school, the way I read papers at the beginning of graduate school was very different from the way I read papers at the end of graduate school or now, right? I'm probably different now, right, compared to 10 years ago too. So I imagine, learning to read primary literature is a lifelong journey, right? For especially for people who stay in academia. So in some ways, yes, I think our students typically would come into the genetics course having some exposure to primary literature, because our introductory biology lab course is a course-based undergraduate research experience where students are engaged in a real research project, and there's some engagement with primary literature in that course. And so then as students come through our curriculum, I wouldn't say that they have a solid handle in reading and understanding primary literature, but they've seen papers, they have looked at them, they have read some of them, and so there's some level of exposure, but I don't think that's a requirement, because I think everybody needs to start somewhere, right? And that somewhere can be anywhere.
Okay, feel everybody keep putting those questions in the chat box. It can be about content related to the paper. It can be questions about maybe a concept was unclear in the paper. Maybe you want Stanley to elaborate on something. So keep coming in the chat box. Until we have more questions about the paper, I'd like to talk a little bit about how to get started in biology education research. And let's say someone took your course in 2017, or maybe hasn't even hasn't taken a course at all. How would you get started without any background in learning sciences?
Yeah, so that's a great question. I think depending on where you are, right, your campus may already have a good number of resources. So for example, if your campus has a teaching and learning center, colleagues there are typically already familiar with the scholarship of teaching and learning, and really collecting evidence in the classroom as a cycle for improving teaching, right? And I know not every campus or university has a teaching and learning center, right? But somewhere on campus, there may be an assessment person who would be doing this kind of work. And I would also really encourage you to connect with colleagues if you have a school of education, right, or a, or a college of education on your campus. In most cases, a lot of those colleagues may be working in K-12 education, right? But the way that they do their research, right, is going to be the kind of approach that we would really do the same, take the same approach, right, in undergraduate biology education, except right the students are a little bit older and the context is a little bit different, right? But engaging colleagues, right, and your school of education on campus, right, that could be a really fruitful collaboration as well. And then and I know colleagues and who do this work, they've also connected with maybe people in math or statistics, right? If you're particularly doing a project that is more statistics heavy for data analysis, right, more quantitative and more statistics heavy. On some campuses, statistics programs, undergraduate programs may have a project-based course where in fact they try to source manageable statistics project from campus and assign them as a project for undergraduate students in a course setting. And those are good places to find resources and colleagues who might be able to collaborate and work together on different kinds of educational projects.
That's great, especially in the time where we're not able to network through societies or have face-to-face conferences. I think that's a great way to start reaching out to people on your campus that might do these things. In the before times, I would suggest meetings like SABER, an ASM conference for undergraduate educators, because these are places where you have scholars who are involved in these studies and you can meet them, ask questions, maybe form collaborations, and form those networks. Unfortunately, we don't have that capability right now. Oh, one person noted in the chat box, the annual meeting for SABER, again, was online this year, and I think you can still access the content. Do you know if that's true?
I think you have to have been a registrant to access the content.
Okay.
But it is true that we are planning to continue to offer some hybrid version of the meeting, at least this coming year, if not in the future as well, because I think the online aspect of it is right. It is really about engaging more colleagues in this work and making the meeting more accessible and affordable for people.
Mm-hmm. Great. I have a question back to your paper. How do you prevent students from looking up the paper? And if you don't tell them the citation, how do you deal with giving proper credit to the paper that you use?
Yeah, so I struggled with that a little bit as well. And I actually give them the citation up front, and Sally and others may have differing opinions. I think they give them the paper at the end, so you still get the citation at the end, right? So I think that sort of I think should take care of the proper credit question. But I actually give them the citation up front, and I sort of tell them like this is about the journey, right? Like if you think about watching a movie, right? You don't want people to just tell you how it ends when you walk into the movie theater, right? So we're sort of going through the journey together, and we're discovering the new pieces as we go along. And I obviously can't prevent them from looking it up, and I think some students do look it up, but I found that for the most part, students, I think they don't, and I sort of reiterate and reemphasize that pretty regularly. So it's mostly based on faith in humanity.
Here's a question for you. Do you have suggestions for designing the CQ or clicker questions for Create? do you feel that there's a formula like scenario, or which of these is evidence?
Yeah, I've done some where some clicker questions where it's right looking at the data, or some clicker questions that's like really great suggestions, right? Like if I write a bunch of sentences, right, which one of this is actually describing the evidence, which one of this is a consistent conclusion. Um, I actually have a clicker question from a different class. It's from the intro bio lab course where we give them it's not even, actual real data. It's like a table of summary of results. But essentially, we gave in four options, right? And of the clicker question, two of those statements are factually incorrect, and two of those statements are factually correct, right? But the two factually correct statements, only one of them is the conclusion to the experimental data shown in the table, right? So in fact, you cannot conclude from the data the other factually correct statement. Um, and, and, and over time, as we've done that clicker question, students are very good at picking out the factually correct options. And then we get like a 40-40 split between the two factually correct statements. And then you get the 10% each picking the incorrect statements. And then they couldn't really distinguish between the two factually correct statement and how to connect it to the data. And so that's been a really great clicker question to really talk about, like how to think through the data and draw a conclusion from it.
That's good. You're really teaching them to think like scientists.
Well, hopefully,
Yeah.
I mean, at least that's the goal.
Yeah. Okay, so let's talk a little bit about group work. It can scare a lot of educators. It has to be very well designed. And how do you motivate your students to make sure that everybody's contributing appropriately? And how do you motivate students to participate in the group work process?
Yeah, I think that's always, a challenge, and I feel like so the way I think about it is, well, when I went to college, right, there's always that row of students at the very back who are reading the newspapers, right, like actual newspapers, right. That doesn't exist anymore, right, but I think those students are still there, and they may or may not be engaged for various reasons, right, and some of them doesn't have to do with the actual course content or the interest in the course, right? It could just be various kinds of life events happening with individual students. So I feel like in a much more interactive classroom, that proportion of students, right, the ones who would have been reading the newspapers in the back, are still there for all those reasons, and they may just be more visible because now they're in a group, and you see them because you're walking up to the groups, right? So I think there's some element of that, right, that could not be avoided. But at the same time, I also think a lot of this has to do with community building, and really getting students to engage with one another as peers and as people before we dive into the much harder intellectual work together. And so we do quite a bit of icebreaker in the beginning. So like the first day of class is actually mostly icebreaker. We actually do, shuffle them, in many different combinations of groups with all kinds of icebreaker questions. So for example, so last winter when we did this, a cognitive scientist on campus actually was developing a mobile app that basically randomly groups students. So you hold up your cell phone, you get a green square, and right, the four students with the green squares would come together, and then when you get together, you get like a bunch of questions pop up on your screen, right, that you talk through about the kinds of hobbies and things that you do. And then I think Gail Heyman, who's a psychologist on my campus, has developed this other app, which doesn't, involve grouping students together, but it's an app called, I think, Beyond Small Talk, where the questions are really designed to be, not personal enough to scare people off in this kind of, first encounter situation, but deep enough that you actually get to know the person a little bit better than just like, oh, what did you eat for breakfast? Or like, what kind of Netflix show are you watching right now, right? But it's a little bit deeper. So we've been, trying to combine those things, and at least, as fall quarter is starting for me now, we're doing a lot more icebreakers on Zoom as well, and try to incorporate some of those elements as well.
Do you ever have students do peer evaluations of how they're performing in the groups and have that count toward their final grade, or is that something that you don't recommend?
We've done, let me think. We've done peer evaluation, not actually, so we've done two mechanisms. And this is not necessarily just this Create course, but I've used them in other courses as well. So one mechanism is a peer evaluation, not of each other, but of the teamwork. And we've done that using the ellipse rubric, which I don't remember what the acronym stands for. Enhancing learning something, and process skills is in there somewhere. Anyway, and it's a group of rubrics developed by colleagues, mostly originating from the POGO project, and so they have many different rubrics related to process skills, and one of them is teamwork, and I've used it in the past for students, again, not to rate one another, but just to say, okay, this is the team. Have you been doing these behaviors, and how do you think you can improve on these behaviors? So that's one mechanism. And then we have done another mechanism where we sort of turn it back on the students as regular adults, right? So if you all agree that there was unequal contribution, then the 3 or 4 of you need to get together and decide what those contributions are, and all of every single person has to sign off. But what you can say is, you got, I don't know, 20 out of 25 right on this assignment. There are 4 of you. You can multiply the 20 times 4, right, to be 80 points, and you can redistribute them. But all of you need to get together and agree on it, and then let us know, right? So that's another mechanism that we've tried to use, and almost never, like, I think over the years of so many groups of students who've gone through these courses that we've taught, we've had like a handful of them request that kind of grade redistribution. And then this quarter, we're also planning to do more community norms at the beginning as part of the icebreaker as well, and to emphasize this kind of sort of thinking about how to include everyone in the group process, but also thinking about how you yourself should, could help hold yourself accountable for equitable contributions, and hold one another accountable for equitable interactions.
Those are great learner-centered strategies that really empower the students to make sure that everybody's held accountable. Sonia, very helpful. Thank you for posting in the chat box the enhancing learning by improving process skills in STEM reference that Stanley was talking about. I was wondering, it wasn't brought up in the chat box, but it's a question I was considering is, are you using this technique in your hybrid or remote classes at this time? And what kind of adaptations have you had to make with not being able to meet face-to-face?
Yeah, so I'm teaching not this genetics course, so I actually rotated off from the I guess 200 level genetics course. I'm teaching a sort of upper division elective genetics course now. I taught it last winter using a more sort of traditional create strategy, and it's on the genetics of pigmentation and how it ties into the social construct of race and racism. So very timely, even though we actually started teaching the course before all the current national discourse. So I am gonna have to figure it out this weekend because classes start Monday on how to do that same course that we did last winter, last January in person that we're gonna do online. So I will let everyone know, or send me an email in December and I can let you know how it goes, how it went. But yes, the plan is to do it, and we're just gonna have to figure out how to do it.
And Stanley has offered, if people have direct questions, you can contact him by email if you wish afterwards. We have a few more minutes if anyone else has any content related questions or questions about doing biology education research, or maybe how to get started with publishing your findings about teaching. You can always ask those kinds of questions too.
Oh, and sorry, one more link. I can find. Let me find that. So the sort of app where students are grouped together randomly. I don't know if that's actually publicly available, but it was designed by cognitive science colleagues here on campus. This is one of the conference papers that they had. I don't know if it's actually available for public use at the moment.
That's a really good thing, because I found when I was teaching, students are really addicted to their phones and they don't want to put it down during class. So embrace it, use it for learning. Good philosophy.
Yeah, I agree. I mean, the phones are not going away. And actually, I think the national statistics is that more students have phones now than students have laptops, I believe. I think some 95, I don't know the exact numbers, but it would be something like 95 or 98% of students have phones, but only like 90% of students have laptops or something like that. The difference is small, but it's a a meaningful difference, I think.
Yeah. And so if you are interested in doing a research study about how your students are learning in the classroom, one way to also get started is making a presentation at a national meeting, such like ASM, Conference for Undergraduate Educators. I'm going to put the link in the chat box, asmq.org. And so there, if you're new to this and talking about your teaching in a scholarly way, then you could give a talk called A Microbrew, which is a short talk talking about your best teaching idea. And that gets you in the door, meeting new people, and talking about your teaching in a scholarly fashion. And as you progress and learn more about how to assess your students' learning, you can go to a poster at ASMQ and go to SABER and start presenting your work. So I think getting started in this work, it's all about meeting people who are already in the field and talking to them about what you want to do and what you want to learn about your students' learning. It's really meeting, networking, meeting people, getting started. And of course, reading articles that are currently published to get up to speed on what methods are being used and what are some of the hot topics. So one question that just came through is, I'm not a biologist, but direct our learning center. And can I publish in JIMBY?
I think that may be Rachel. Would you like to? I mean, the short
Yeah.
answer I think is yes, but Rachel, would you like to
Yeah.
weigh in?
So if you're assessing learning about biology broadly related, then I think that GIMBY would be a great place to go. And if you have further questions, Martha, you can email me directly, and I'm going to put my email in the chat box. And here's the nice thing about GIMBY is that they have a suite of editors that are more than willing to work with you. If you send them an abstract, they can give you feedback on the section that's most appropriate for you before you do a full-scale manuscript ready to submit. Okay, so always ask in advance, email me, email senior editors, and we're willing to help you out along the way.
Rachel, I think there's another question about Jimby in the chat.
Okay, so, oh wait, thank you for pointing that out. Is Jimby interested in scholarly works and innovative models of student and faculty development programs? And is Jimby interested in scholarship around advancement of underrepresented minorities and other marginalized groups? Would you like to take that a little bit?
Yeah, I'm tangentially familiar with Jim B, right, as an author. I think Jim B had a recent issue dedicated to equity and inclusion, a special issue, and what's the other word? Scholarly works and innovative models of student and faculty development programs. I think I've seen articles in those areas previously in JIMBY as well. There are different article types, right? There are research articles, which I think, a wide range of research topics would fall under. And then there are, I don't remember the formal name where it's more curriculum materials, right? And then there are perspectives and essays and various things. So I think, especially for the research articles, right? I think anything that would fall under microbiology and biology education would fit with the journal as well. I don't represent the journal, right? So that's at least my experience as an author. And I don't know if Rachel from the ASM side, you have more insights into this.
Yeah, you're spot on. And thanks for giving us the end user perspective. Jimby is definitely interested in scholarly works and models of student and faculty development. So for instance, one question would be, how do we encourage faculty to use inclusive teaching, right? So that would definitely be of interest. And also Jimby's interested in scholarship around helping underrepresented minorities and marginalized groups and STEM. So any program that you might do research about that helps students achieve learning outcomes or achieve success in STEM would definitely be welcome. So yes and yes is what we were getting at. And there's different types of articles in Jimby, as Stanley was alluding to. His paper that Stanley talked about today was a research article. And so that's hypothesis-driven research where you have a variable and you test it, and you have assessment data. A lot of scholars that are new to publishing biology education research may publish a shorter 800-word tips and tools article that describes your best idea for teaching biology or microbiology, and no assessment is needed for that. So, and if you want to learn more, you can always email me directly about the types of articles. And there are publication fees in Jimby in order to support the journal and its open access model. And the information about the fees is posted at the Jimby submissions website.